It's a pleasure to be here again with you to present more one webinar about bio-waiver regulatory cases from Latin America. Today, October 24, we have the participation of a Brazilian specialist, and we'd like to present today about the new legislation that was implemented this year. So before starting this webinar, I would like to present myself. I'm Thalita Ferreira. I'm a Brazilian pharmacist with more than 15 years of experience in pharmaceutical companies in Latin America. I have an MBA in Innovation in Health and a master's degree in entrepreneurship. And at the moment, I'm doing my PhD in economics from the University of Salamanca in Spain in collaboration with the University of Health and Science Pharmacy in St. Louis, Missouri, in the United States. Also, I am a coordinator of this project for VIP in Latin American region. So I would like to share with you some announcements that is important to know about this webinar. This webinar is recording and you can access at YouTube platform in the channel of VIP. All the information that you would like to know more about the events that VIP is conducting, you can access our website. And also, if you have any questions, any suggestions, you can send me while we are conducting this webinar by our chat box, but also you can send any information to our email. And at least, but it's not less important, I'd like to tell you to become a member of a VIP. So make your inscription at our website, and you can understand more about how we can work together to transform the pharmaceutical environment. I'd like to also to thank you to a stern commitment of this project that we have in Latin America, Dr. Giovanni Paulette, Dr. Alex Aceituno Alvarez from Chile, Andres also from Chile, Pablo also from Chile, and Dr. Miguel Angeles Cabrera from Cuba. Thank you very much for supporting this project, working together to transform our region and also sharing knowledge, right? So thank you very much for external partners to support this project and to make it happen. Today, we have the participation of Eduardo Agostinho Fernandes from Brazil. Eduardo is graduated in pharmaceutical science from the University of Minas Gerais. He has specialization in surveillance from Fiocruz, and also specialization in pharmaceutical technology from Fluminense Federal University from Brazil, and a master's degree in health and science from University of Brasilia, and where he had the opportunity to present the analysis of factors that interfere in the results of bioequivalence studies of medicines in Brazil. Since 2005, Eduardo is a coordinator of equivalence studies at Anvisa, and since the beginning of this year, he agreed to participate of a member of Latin American bio-waiver commitments that we have this project support by VIP and the WHO. Thank you very much, Eduardo, for your participation and to be here with us. I will share our other screen with your presentation. Just a minute.
Okay, thank you, Thalita, and thanks all to FIP for the invitation to share the information regarding the ADC 749 that was just published. So during this presentation, I will cover the main aspects of the RDC 749. That this resolution was published on September 5 and became effective on October 3. And in this resolution, we cover the main aspects regarding the biowaivers. Next, Talita, please. So the RDC 749 is divided in three main sections. The section 1 is related to biowaiver by pharmaceutical dosage forms. The section 2 is for biowaiver by dose strength. And the section 3 is biowaiver by bioclassification system. We are going to cover the main aspects of each section of this resolution. Next, Talita, please. And the first one is the section 1, of course, that is related to biowaiver by pharmaceutical dosage form. Next. So when we say the word pharmaceutical dosage forms, we mean that according to the pharmaceutical dosage form, the medicines can be biowaived. So it means that there's no need to conduct a bioequivalence study according to the pharmaceutical dosage forms, and this is applicable for oral solutions. And there is also there is of course a concern regarding this excipients. So in this case, the excipients should be qualitatively identical and quantitatively similar regarding the comparator product that is usually the reference medicines. And this is also applicable to aqueous and oil solution for parenteral use. And the same concern regarding the excipients is applicable. And for oral inhaled medications as nebulizers, nasal sprays, aerosol. So for the Anvisa's regulation, we have a specific RDC, a specific resolution, that is the RDC 278, that describes better how to perform and when it is necessary to perform some in vivo assessments of the formulation. And for ophthalmic aqueous solution, it is also possible to apply the biowaiver by pharmaceutical dosage form, but in this case, there is a concern regarding the excipients present in the formulation. And for oral solids intended for local action, it is possible to apply the biowaiver by pharmaceutical dosage form, but it is possible to have some necessity of perform some additional tests. In this case, it is not easy to describe in the resolution when it is necessary to perform some additional in vitro tests in order to apply the biowaiver by pharmaceutical dosage form. So it is in this case it is necessary to send a consultation to Anvisa in order to this answer to be given. Next, please. And biowaiver by pharmaceutical dosage form, it is also applied, may be applied, of course, by aqueous ES solution with a concerned regarding the excipients present in the formulation. And for pharmaceutical forms for topical application not intended for systemic effect, the biowaiver by pharmaceutical dosage forms may be applied, But in this case, the sequence should be the same and in the same amount and within the same physical, chemical, and micro structural behavior compared to the comparator product. Talita, please. Next. Now moving to the section 2 that is related to the biowaiver by dose strength. This kind of biowaiver is applicable to immediate and modified release drugs. So it can be immediate or modified release drugs. But for modified release drugs, the drug should have the same mechanism of release from the pharmaceutical dosage form in all strengths that are trying to be biowaived. So it cannot be a different mechanism. And also the formulation and also all the strengths should be manufactured in the same address, so in the same site, which and this requirement is not applicable for the BG8 release drugs. And how can we I think how can we come to the conclusion that we should conduct the bioequivalence study with the higher or with the lower strength or both? So it depends on the proof of the linearity of pharmacokinetics, the proportionality among formulation. This proportionality is regarding the oh, can you hear me now?
Yes, I can hear you.
So Talita, during the presentation of the previous slide, were you able to hear me? Oh.
Yes. Okay, everything's okay.
Great.
Okay, I believe that's just a cut, but everything's okay now.
Okay, great, thanks. So continuing this topic of biowaiver or by those strength, regarding the linearity of PK. PK is a pharmacokinetics. This is a feature related to the drug, so it doesn't depend on the formulation. And when when we can see that the PK is linear for the drug, the bioequivalence study should be conducted with the higher strength. For example, if I have a 10, 20, and 30 milligrams strength. So in this case, we should conduct the bioequivalence study with the 30 milligrams and the 20 and 10 milligrams formulation can be biowaived. But if there is a concern regarding the safety of the subjects, while having the 30 milligrams formulation, it is possible to conduct the bioequivalence study with the 10 milligrams, and then the two higher strengths can be biowaived. But if the pharmacokinetics pharmacokinetics is nonlinear due to saturation of the drug biotransformation, it is necessary to use the higher E strength in the bioequivalence study. This is because in this case, the higher strength is considered to be the most sensitive strength to be used in the study, as if the test and the reference products are not bioequivalent in the highest strength, this difference will be much more visible. And when the PK is nonlinear due to saturation of the absorption process, so in this case, it is necessary to use the lower E strength in the bioequivalence study, and then the higher E strengths could be biowaived. And when the pharmacokinetics is nonlinear due to the lack of solubility of the drug, so in this case, it is necessary to conduct more than one study using the higher and the lower strengths. So in this case, at least 2 studies would be necessary. Next, please. And now I'm going to cover the section 3, that Biowaiver by Bioclassification System. I would like to highlight that the section 1 and 2 we did not have many changes considered the previous resolutions, as the RDC 37 of 2011, the rules for the bioequivalence by those strength are basically the same, and there was some kind of improvement in the excipients criteria for the Biowaiver by pharmaceutical dosage form, comparing this new resolution and the previous one. But for the section 3, that is Biowaiver by Bioclassification System, we have we had some changes. But although for Anvisa, it was possible to apply the BCS Biowaiver in the past, but only for a list of some drugs that we have described in a list that we called a positive list. Nowadays, we are open for any drug that can be proved that the classification, the BCS classification, is class 1 or class 3. And so the BCS Biowaiver is applicable to class 1 and class 3 drugs. Yeah. It is also applicable to prodrugs only if the prodrugs is absorbed as a prodrug. So there is no possibility to have some changes in the drug before the absorption. And it is also applicable to drugs for oral use for systemic effect. It is not applicable for the drugs with a narrow therapeutic index that we usually call NTI. It is not applicable for drugs with the absorption in the oral cavity. For example, the tablets that we usually place under the tongue for fast absorption. So this is not the case, and this is not applicable for modified release products, and also not applicable for products not administered with water. Next, please. And it is applicable for fixed dose combinations, but in this case, all the drugs, at least the 2 drugs in the same pharmaceutical dosage form, should meet the BCS criteria. Yeah? And so for the application of the BCS biowaiver, we need to know the classification, the BCS classification of the drug. So it means that we should be able to perform the determination of the solubility and also the permeability, and this is going to allow us to know the bio classification of the drug. And how do you perform the determination of the solubility? So in this case, the highest therapeutic dose should be soluble in 250 ml of aqueous media in the pH 1.2, 4.5, and 6.8. This experiment should be conducted using the equilibrium solubility experiment, and that is usually related to a method named shake flask. So you need to perform a continuous agitation of the solution and collect some samples and quantify them. Then during the time, in order to reach the equilibrium, and during the experiment, the pH should be measured, and as the pH should be kept in the pH 1.2, 4.5, and 6.8. And during this experiment, the stability also the drug should be verified, and if it is known that it is verified that there is a degradation of more than 10%, so we assume that this solubility cannot be determined. So we cannot have this number as this number of solubility in order to come to a conclusion for a bioclassification system. So and of course, the quantification method should be able to differentiate the drug and the degradation product that can be can appear during the experiment. And of course, the quantification method should be validated. Next. And for determination of the permeability, the other feature that we need to know in order to come to a conclusion of the bioclassification of a drug. So we can say that when the absolute bioavailability is higher than 85%, or the recovered of the administered dose unchanged in duodenum is higher also than 85%, we assume that the drug has a high permeability. And this information, when available, is usually available from the scientific literature. Also, some in-house studies can be used to provide this information. And also, it is important to highlight that the stability data of the drug in the gastrointestinal tract may be necessary, and in this case, the degradation may not be higher than 10%. But when this information regarding the absolute bioavailability is not available, it is possible for the sponsor to perform an in vitro test using a method using a Kakocho cells. And we are going to talk a little bit more about this kind of method. Next, please. So Anvisa, along with the IDC 749, Anvisa has published the Instrução Normativa 182. It was published on the same date. And in this Instrução Normativa is described some details of the Caco-2 cell permeability experiment. Caco-2 cells are nothing more than some kind of human colon adenocarcinoma cells that are placed in a platform, and we use this layer of cells in order to check the amount of drugs that come across from the apical part, the upper part of the cells, to the part to the bottom side of the cell. So this is a kind of permeability experiment. Next, please. And for these, the permeability by in vitro methods using Caco-2 cells, the method should be validated using at least 5 drugs with known permeability, right? And for each of these drugs, it is necessary to have at least 3 replicates. And the standard of drugs that we have are known to have a 0 permeability. For having a low permeability, that should be a drug with less than 50% of permeability. A drug with the intermediate permeability, which is considered to have from 50 to 84% of permeability, and a drug with high permeability, which is considered to have more than 85% of the permeability. It is in doing the validation, it is necessary to differentiate drugs with low, moderate, and high permeability. So it means that during the validation process, all of these characteristics of these drugs used during the experiment should be confirmed. And so it is also necessary to have a quantification method in order to quantify the amount that come across that the monolayer of cells. And the integrity of the Caco-2 cells should be confirmed. And one way of doing this is using this technique named transepithelial electrical resistance, ET. And also the 0 permeability drug can be used to confirm the integrity of the Caco-2 cells monolayer. Next, please. And regarding the permeability test itself, it is necessary to access the drug efflux from apical to basolateral. It means that from upper part to the bottom part of the monolayer of Caco-2 cells, and also from basolateral to apical, from the bottom part to the upper part of the monolayer Caco-2 cells. This test should be conducted using in the same conditions of the validation. And doing the experiment, it is necessary to add a higher and a moderate permeability drugs in the apical compartment. It's going to be an internal standard during the experiment. it is not possible to have some incompatibility between the drug under investigation and also in the internal standards for reasons that it may interfere in the result of the experiment. And the test drug is considered highly permeable when the permeability is equal to or greater than the high permeability internal standard. Next. And the last set of data that should be submitted along with the application is the dissolution profile, in order to verify some information regarding the formulation. So the batch used in the dissolution profile should be representative of the manufacturing process. And the comparison of the dissolution profile is done using the rules described in the RDC 31. So is usually the F2 method is applicable for doing the comparison of the dissolution profile. And in this case, the dissolution profile experiment should be conducted using some specific conditions. So this is not possible for the application, for the applicant to develop its own dissolution profile method. So these conditions should be used. So it is necessary to use the apparatus pedal 15 RPM or basket with 100 RPM. The media used should be with the pH 1.2, 4.5, and 6.8. The temperature of the media is should be 37 plus minus 1 degrees Celsius, and the volume used should be in the maximum 900 ml, and no surfactant is allowed to be added in the media. Next, please. And for BCS class 1 drug, the requirements for the dissolution profile results is 85% in 50 minutes or 85% in 30 minutes. For BCS class 3 drugs, the requirement is 85% dissolved in 50 minutes. And it should be conducted one dissolution profile for each strength of the medicines. For example, if I have 10, 20, and 30 milligrams tablets, I should be able to conduct the 3 dissolution profiles of pH 1.2, 4.5, and 6.8 for each strength, 10, 20, and 30 milligrams, and the results should be as this one that I have already talked about. Next, Clarita, please. And the last topic relates to the biowaiver by BCS is regarding the recipients. So in this case, the formulation, the test formulation should mimic the comparator drug product, which is usually the reference product. And if there are differences between formulations, it is necessary to perform the assessment of a potential impact in vivo. So this justification should be also presented with the submission of the sponsor. For BCS class 1 drug, qualitative and quantitative difference are allowed, except for excipients that affect absorption. So the limit is 10%. And for BCS class 3 drugs, the qualitative and quantitative excipients should be the same. And when we mean the same, we mean that some a limit of 10% of difference in quantitative in terms of excipients in quantitative way is allowed. So it is in maximum 10% change in difference between formulations. And I think I could cover the main aspects of the resolutions 7449 that we are trying to apply here in Brazil. We haven't received any submission so far regarding these the rules described in the RDC 749, but we're expecting to receive it soon. And the main goal of this presentation was not to cover all the details of the resolution, but only the main aspects. But if you have any questions, I will be glad to have it. Thanks a lot.
Thank you very much, Eduardo, for your presentation. It was very good, and I believe that we have some questions at the chat box that we can answer. So if meanwhile, Eduardo, you answer the questions that we have in the chat box. If you have any more information, or if you need to know more about this project, feel free to contact us. I will put my email here that you can contact VIP and understand how we can support and sharing knowledge in our region. So, Eduardo, we have some questions in the box, in the chat box. So the first question that you have, it is about the bio-waiver. So Cassim sent a message, will bio-waivers be considered for suspension for oral administration?
So the answer for this question is no. Suspension is not a pharmaceutical dosage form that could be bio-waived by the rules described in the RDC 749. So in this case, it is necessary to conduct the bioequivalence study.
The next question that we have, it is Assad. So it means that bioequivalence is majorly essential for oral solid dosage form, which are low soluble and complex products.
So the answer for this question is a little bit more complicated. So I think that the biowaiver for BCS classification is only an alternative of proving the safety and efficacy of a drug. But so as I described during the presentation, it is necessary for the formulation to fulfill lots of requirements, not only a drug requirement. But in this case, even if we have a BCS class 1 or class 3 drug, it may be necessary to conduct a bioequivalence study when the other requirements are not fulfilled. And of course, the bioequivalence is the main tool to prove safety and efficacy for the formulations that we consider as a complex one, or for a very low soluble drug.
Okay, thank you. Perfect. So now, Alex Azeitona, I believe that you'd like to talk, right? Not just write down your question. Are you allowed you talk? So Alex
Are you
Aceituno,
listening to me?
yeah, yes, we can.
Okay.
Alex Aceituno is from Chile, right? He's from a health authority agency in Chile. So Alex, go ahead with your question.
Thank you, Talita. I have a couple of questions regarding the very nice presentation you made. I was thinking through your presentation, and I got the feeling of white you decided to use for the solubility, the highest therapeutic dose, when in reality, when we perform the BE studies, I mean in vivo studies, you use the highest dose strength. So it looks like to me that there is no like a real, how can I say it, real reason for the forcing a situation that it's clinical, mixing with something that it's real technical. I mean, this solubility is a technical aspect of, in this case, of the drug. The therapeutic use is a clinical thing. It's not related to completely to what we are talking where we can perform in vitro dissolution or getting a biowaiver, but BCS or whatever the reason is. This is one of my first questions. The second one is the why. I'm not. Yeah, that's not the question. Is it possible for ANVISA to think to get some different setup for permeability studies instead of CACO2 cell? Because as long as the method it's validated, I don't see any reason for not accepting some additional setup, like working with different cells apart from CACO2 cell. Those are the main question. I just they came to my mind, and I would like to share with you and see what is the background I can receive from you. Thank you.
Thanks a lot, Alexis, for your comments and questions. Well, for the first one, I did not mention this information during the presentation, but when the highest therapeutic concentration of the drug is not soluble in the 250 ml of each pH of that, it is possible to present a justification, and for using only the amount of drug that is contained in the highest therapeutic dose pharmaceutical form. So it is possible also to provide this information with the justification. So the RDC 749 allow this kind of approach. And for the use of cells, other kind of cells, not only the CACO2 cells, for the permeability experiment. The RDC focus only the CACO2 cells. So in this case, in this moment, it's not possible to use any other cells approach for presenting information regarding the permeability of the drug. So we focus only on the CACO2 cells. I believe because it's a cell model using human cells. I believe that there are other human models, other cells models that are not using human cells, as MDCK and others. But I believe that in this moment, we focus only in human cells, as we are. This is the first approach that we are assuming that it is possible to have an in vitro permeability study to come to a conclusion regarding the permeability of the drug. I believe maybe in the future we may have some other alternatives than only CACO2 cells.
Thank you, Eduardo. Just a quick question, an additional one.
Okay.
Is it possible for the for BCS application? I'm not sure if it's the case in Brazil, but suppose you have two dosage form that they have different salt. I would say sodium and maybe potassium, different salt in terms of a comparison. Would you accept that as a possibility to get a biowaiver by a BCS system?
Not in Brazil, but it is not possible, not because we can see some technical information that we cannot allow this. It's more because it is related to the concept of generic products in Brazil. It is necessary to have the same salt. So as a biowaiver is only a tool to prove safety and efficacy of a generic formulation, all of the requirements for generic is the same, and we have only one additional tool. But so in this case, it is not possible because of the resolutions and regulations that are in the background of this one.
Okay, and one one last thing. Sorry,
Okay.
Is it possible for generic Brazilian companies to use because you were telling me during the seminar that it's the formula needs to be very similar, either quantitative or qualitative similar. So is it possible for generic companies to use the reverse engineering at this point? Is it allowed?
Yes, of course. Actually, we're expecting this to happen because as the formulation of the test product should be very similar to the comparator. The only way of doing this is perform the reverse engineering. So it is possible. Yes, the answer is yes.
Excellent. Thanks, Eduardo.
You're welcome.
Thank you very much, Alex and Eduardo. So it's so great when we can align the information about the new legislation and to go ahead with new implementation to support pharmaceuticals in our region, right? So I'd like to finalize this webinar, telling to everybody that all webinars that we conduct here are available at events.vap.org. So everybody can access this webinar that we conduct now, but others that we have already conducted in the past, and the next one. So next month, we have a webinar with Colombia, the health authority from Colombia, that you present their new legislation that they have there for biowaiver. And in December, we will have a webinar with Mexico. So if you wanted to receive information about our webinar, contact us. So here it is my email. And thank you very much for your attention and participation. Thank you very much, Eduardo, for your time and sharing your knowledge with everybody. And we keep in touch, and you'll see you in the next webinar that you conduct in next month. Thank you very much, and have a great week for everybody.
Thank you. Bye.